Welcome, everyone. We're going to just wait a couple minutes for people to be let into the Zoom. Okay, let's go ahead and get started. Welcome to this webinar for December. I'm Christy Sullivan. I'm the Secretary of the American Society for Cellular and Computational Toxicology. This webinar is presented by ASCCT, the Japanese Society for Alternatives to Animal Testing, JSAAE, and the European Society for Toxicology in Vitro. I want to encourage you to visit all of our websites to find out more about our organizations and consider becoming a member to support programs like this webinar series. Good morning, good afternoon, good evening, wherever you're joining from. Hope you're having a good start to your holiday season. So just want to encourage you to join our mailing list for job and event announcements and let you know a couple of housekeeping details. The webinar is recorded and the recording will be posted on the ASCCT website a few days from now. In the webinar platform, you can ask questions of the presenter using the Q&A feature in the toolbar at the bottom of your screen. There will be a Q&A period after the presentation is finished. You can add your questions at any time, and other participants can see and vote for questions they really want to hear the answer to. We've also enabled closed captions, so you can turn them on or off in the toolbar according to your preference. And then finally, please say hello in the chat and let us know where you're joining from. Just a couple of quick announcements before we get started. First of all, we want to thank everyone who attended or spoke at or supported or helped to plan the ASCCT annual meeting. We just held it in Gaithersburg at the IIVS headquarters. As many of you know, it's been a challenging year for federal scientists and federal agencies, and we were just very happy to have still have some of our plenary speakers, including Tracy Beth Hogue, be able to speak. And some of our federal attendees were not able to attend, of course. And so we will be holding some webinars for the missed sessions, and they will be open to everybody. So stay tuned and sign up for our mailing list if you want to learn about when those will happen. And finally, if you want to join the organizing committee for next year, please get in touch with us. I want to also remind you that ESTIV, it's having its annual Congress in Maastricht in June 2026. So please visit their website to find out more about that. I believe abstracts are closed at this point, but stay tuned in case once registration opens. We have several webinars that will be coming up next year. These are going to be annual meeting related presentations, as I just said, and also award winner webinars, presentations of work that won awards at the ASCCT annual meeting or the JSAAE annual meeting. We'll organize those webinars in 2026. So this webinar is now a December tradition. It's our third time that we're having a joint webinar between JSAAE, ASCCT, and ESTIV, where JSAAE members select the speaker and bring their work to the ASCCT ESTIV webinar series to present it to us. And I'm happy to have Dr. Yasu Kanda, Vice President of JSAAE, here to say a few words about their activities. Yasu?
Yeah, thank you, Beth, Christy. I'm Yaskanta from NIHs. And as Christy said, so JSAAE is now working with STIB and ASCCT under a memorandum of understanding to collaborate together to promote 3Rs and NAMs globally. And early career researchers, Kaneko-san and Rai-san, received the STIB awards at the JSAAE annual meeting this year. And JSAAE members also participate in the ASTT and STIB meeting every year, as you can see the right panel. And next slide, please. And in terms of JSAAE annual meeting, we have 946 participants featuring 2 educational lectures and 20 symposia this year. And as you can see the right panel, so high school students also presented posters highlighting our society's commitment for education for next generations. And importantly, the international session at the annual meeting featured invited guest speakers from Asian countries, from China and South Korea and India, who delivered a wonderful presentation on NAMs. So we JSAAE look forward to strengthen our collaboration with international partnership and working together to advance NAMs and globally. Again, thank you, Beth, for having a great opportunity to collaborate together. Thank you very much.
Thank you so much, Dr. Kanda. We're really excited to hear more about this work and to keep the partnership going. So today's presentation is development of in vitro evaluation model for drug-induced proximal tubular injury using human proximal tubular epithelial cell spheroids. It's presented by Dr. Hiroshi Arakawa, who is a professor in the Department of Regulatory Science at Nagoya City University Graduate School in Pharmaceutical Sciences. He holds a PhD, an MS, and a BS, all in pharmacokinetics. His research and ongoing work include construction and development of biomimetic models for drug safety assessment, drug side effects and drug-drug interactions via drug transporters, elucidation of mechanisms involved in pharmacokinetics and safety using clinical data, and elucidation of dynamics and physiological effects of uremic toxins. Dr. Arakawa, the floor is yours.
Thank you for a kind of introduction, Christian. So I'll share my presentation. Can I see my presentation?
Yeah, it looks good.
Okay, so I'll start my presentation. Thank you very much. Okay, I really appreciate this nice opportunity to organizers. So I'd like to talk about the development in vitro evaluation method model for drug-induced proximal tubular injury using human proximal tubular epithelial cell spheroid. So acute kidney injury was caused by exposure to chemicals, including drugs, which significantly affects patient health and prognosis. 20% of clinically observed kidney injury are caused by drug treatment, and its development has a major impact on the pharmacotherapy. So therefore, in drug discovery and development, animal testing is used to evaluate kidney injury during preclinical research. However, the prediction rate using the experimental animals, such as rodents and dogs, is less than 50%. The specificity is relatively high. So it is desirable to establish a highly predictive evaluation system using human kidney-derived cells. The kidney consists of nephrons, composed of the glomerulus, proximal tubular and loop, and distal tubule and collecting ducts. Clinical reports have identified the drugs and biomarkers that exhibit the site-specific toxicity within these individual segments. Among the various cell types that make up the nephron, proximal tubules here is most frequently observed in drug development and has become a major area of interest for pharmaceutical companies. So one reason drug-induced toxicity tends to occur in the proximal tubules is that this segment receives a high proportion of renal blood flow and contains a large number of mitochondria next to the cardiac muscle. So in addition, the epithelial cells, RPTEC, proximal tubular epithelial cells, RPTEC, has abundant drug transporter expression, such as organic cation transporter 2, organic anal transporter 1, OAT3, and PZP. These transporters that actively uptake circulating compounds into the RPTECs, causing the drug-induced kidney injury. For example, cancer drugs, cisplatin was taken up by the OCTs efficiently into the RPTECs and act the mitochondrial injury, and then so causing the DIKI. Furthermore, several antivirus compounds, nucleoside analogs, was taken up by the OAT1, OAT3, and then so causing the cytotoxicity. Thus, drug transporters play a crucial role in the development of drug-induced proximal tubular injury. This figure summarizes the protein expression levels of major drug transporters obtained from proteomic analysis of the human renal cortex, which contains the proximal tubule. It clearly shows that transporters known to play a crucial role in renal drug disposition, such as OCT2, OAT1, MATE1, OAT3, and PGP, express high absolute levels among the renal drug transporters. Furthermore, this table summarizes the clinically important drug substrate and inhibitors for each transporter. For example, so OAT1 efficiently transports the nucleoside analogs antiviral drugs such as aciclovir, tenofovir, and zidovudine, cidofovir. Furthermore, OAT3 transport organic annual compounds of relatively higher molecular weight compound compared with OAT1, such as bumetanide, methotrexate, and furosemide. Furthermore, so OCT2 transports the cisplatin as well as the oxaliplatin and metformin and pindrol. So this transporters may play an important role in the drug-induced kidney injury. A representative example of illustrating the impact of drug transporters on the drug-induced reproducibility is the involvement of OCT2 in cisplatin-induced kidney injury. This is the OCT2 knockout mouse study. Cisplatin treatment to the wild-type mice increases the renal kidney injury biomarker BUN. So, but in the OCT2 knockout mice, BUN increase was attenuated. Furthermore, cisplatin accumulation in the kidney is markedly reduced in the knockout mouse compared with wild-type mice. Consistent with these findings, so clinical observations in humans have shown the patient's functionally impaired OCT2 variants exhibit a slight increase in the renal injury marker creatinine following cisplatin administration, compared with patients with GG genotype, wild-type genotypes. So, drug transporters may play a role in the drug-induced kidney injury. So, as a human proximal tubular epithelial models, So immortalized cell line HK2 and primary culture of human RPTECs are commonly used. However, these cells have major limitations. One is their expression levels of transporters involved in the drug disposition are low compared with the renal cortex. Sorry, the open bar is the renal cortex and the black bar represents the HK2 cells transporters. Furthermore, immortalized cell lines predominantly generate ATP through glycolysis rather than through the mitochondrial oxidative phosphorylation. This non-physiological metabolic state results in the diminished physiological responsiveness and limits their ability to reproduce the drug-induced kidney clinical injury. So, when construct in vitro evaluation system for drug-induced kidney injury, so we focus on the three-dimensional culture of RPTEC spheroid, like here. In this spheroid, we have shown that the OAT1, which is a lower expression in the conventional 2D culture, has the high expression levels close to the human kidney cortex. And there is a very good reproducibility in the product lot and donor lot. So as long as OAT1, so improved expression of 5 major drug transporters such as MATE1 and PGP. So furthermore, so the spheroid able to culture the maintaining OAT1 expression over 40 days. And by the proteome analysis, so protein expression of 71% of membrane transporters are within 5-fold of human renal cortex. So physiologically, so RPTECs primarily generate ATP through beta oxidation of fatty acids. Therefore, we first characterize the energy production profile of this cell model. Various inhibitors of ATP synthesis were added to the cells, and the intracellular ATP levels were measured. As a result, so this is the etomoxia beta oxidation inhibitor. Etomoxia did not reduce the ATP levels, but the phosphorylation inhibitor, rotenone, markedly reduced the ATP levels in the 3D RPTECs. And 2-deoxyglucose, this is glycolysis inhibitor, 2-deoxyglucose and IAA was markedly reduced the ATP levels. This results, the extent of the ATP reduction induced by the rotenone is greater in the 3D culture system compared with 2D cultures. These findings indicate that the 3D RPTECs mainly produce ATP through the aerobic respiration using glucose, not fatty acids. So to evaluate toxicity associated with long-term drug exposure, so we set the culture period to the 28 days and examine the characteristics of 3D RPTECs over the duration. So throughout the 28-day culture period, so no noticeable change of cell morphology and mRNA expression of OAT1 and OCT2. And this expression was higher compared with 2D RPTECs. Sorry, missed the number of the slides, so okay, I skipped the presentation. So, furthermore we investigated the characteristics of DI drug-induced kidney injury evaluation using 3D RPTECs. The OCT subset cisplatin and OAT1 subset tenofovir were exposed to the 3D RPTECs until the 28 days. As a result, so decrease in ATP in the 3D RPTECs culture system even at the decrease even at the concentrations whose no toxicity was observed in the 2D cultures. in using the cisplatin and tenofovir. So this system can evaluate the transporter-mediated drug-induced kidney injury. So to determine whether transporters contribute to the ATP depletion in the 3D RPTECs, we treated the cells with the cisplatin or tenofovir together with the inhibitors of OCT2 and OAT1 inhibitors, simethicin and probenecid. So as a result, co-administration of simethicin, OCT2 inhibitor, attenuated the cisplatin-induced ATP reduction. And similarly, the OAT1 inhibitor probenecid reduced the ATP depletion caused by the tenofovir. So these findings demonstrate that 3BRPX model can be used to evaluate drug-induced toxicity mediated by the drug transporters. Then, so we further characterize the evaluation ability of 3D RPTECs using the about 30 kinds of drugs, which is the presence of absence of expression of proximal tubular injury in clinic. So after exposure to drugs, so we measured the intracellular ATP levels by a cell titer growth reagent and calculated the 20% decreasing concentration of ATP, and then so divided by the clinical Cmax values. So drugs were selected with the pathologist and referring the papers investigating drug-induced kidney injury by Pfizer and Takeda corporations. So we chose the 17 kinds of positive drugs for proximal tubular injury in clinics. For selecting drugs, so we considered the toxic mechanisms. For example, so the drug transporters and mitochondrial organelle injury. So and then, so we chose the negative compounds. This is more important. So we chose not only the non-active compounds such as the mannitol and vitamins, but also drugs representing the toxic activity in other than the proximal tubula. For example, doxorubicin here is expressed podocyte injury, but not in the proximal tubule. So we set as negative compounds. Furthermore, ciprofloxacin is caused the liver injury and crystal nephrotitis in the collecting duct. So, but they don't express the proximal tubular injury. So we set as negative compounds. Furthermore, beta blockers and ACE inhibitor express the cause kidney impairment by reducing the renal blood flow. However, so because they don't exert direct cytotoxic effects in the renal cells, so they are classified as a negative compounds in this study. So I described our literature with details. Please refer this literature. So we investigated the concentration-dependent assay for 7 and 28 days and calculated the EC20 values for the ATP reduction. Black and red circles represents the 7 and 28 days exposure study. So we set the 100 of the ratio of the EC20 and Cmax value. We set the 100 as a criteria. The sensitivity of the positive compound is about the 82.4% and the specificity is 100% at the 7 days. So, and as a false negative drugs, cyclosporin A and tacrolimus were observed at day 7. And when we investigate for the 28 days exposure test, cyclosporin A was judged as a positive compound, but the specificity was reduced in the 3 compounds. Therefore, sorry, okay. And then, in addition to benchmark the performance of this cytotoxicity assay, we conducted toxicity studies using the 2D RPTECs and immortalized HK2 cells. So as a result, 3D RPTEC system successfully detected transporter-dependent cytotoxicity and showed no false negative among the drugs tested and demonstrated the highest AUC curves, AUC values compared with 2D RPTEC HK2 cells. So 3D RPTECs demonstrates the highest predictive performance among the models evaluated. Since evaluating the intracellular ATP measurements demonstrated the high predictive performance in 3D RPT model, ring study was conducted for an evaluation of technology transferring and reproducibility of experimental results. The study was conducted in which 6 domestic Japanese manufacturers, including 3 pharmaceutical companies, 1 food company, and 2 CRO participated. The tested compounds were purchased in the bulk at the Canada University. This is my previous institute, and distributed to each company without branding. In this study, 4 compounds were used as a positive compound: cisplatin, cephalotin, vancomycin, and adefovir. And 1 negative compound was selected, mannitol. To enhance the reproducibility of the experiments, we prepared the instruction manual and unified exam recording paper, Excel file, Excel sheet. And then, we started the ring study. The results of 2 tests conducted at the 6 companies are shown here. This is about the 12 test assay from the 6 companies. As shown here, high intra-institutional reproducibility was observed in the 5 compounds, 4 positive compounds and 1 negative compounds in the DIK evaluation study. Now, we have, I think these are very good results. We are now planning the international ring study now. On the other hand, the cytotoxicity assessment based on the intracellular ATP levels produce favorable results for the many compounds. However, cyclosporin A did not affect the ATP levels, but the cell morphology slightly changed. Therefore, we proceeded to evaluate additional markers beyond the ATP. In this study, the renal injury markers shown here are TGF-beta, MCSF, clusterin, KIM1, interleukin-1 beta, 18, alpha-B, TFF3, and HO1 were chosen, and here are measured using the ELISA system. Because kidney injury can arise not only from the cell death, but also from toxicity mechanisms such as fibrosis driven by the secretion of inflammatory cytokines. We selected these cytokines. And furthermore, HO1, which responds to the intracellular oxidative stress and the hypoxia, and also measured. So as a result, exposure to 4 nephrotoxic compounds, cyclosporine, omeprazole, rifampicin, and vancomycin, led to a marked increase in intracellular HO1 levels. In addition, cyclosporine A, which induced renal fibrosis, triggered detectable increase of TGF-beta and MCSF. This is a macrophage stimulating factor. So these findings suggest that in drug exposure studies using 3D RPTECs, quantifying inflammatory markers in addition to the intracellular ATP levels enables the characterization for underlying toxicity mechanisms. So in addition to further characterize the drug-induced toxicity, we conducted high-content analysis using organelle-specific markers. Which is methods can detect the changes in the multiple cell phenotypes from the same cell by staining with fluorescent probes, such as a mitochondria, which represents the mitochondrial membrane potentials, and cellulose greens, deep red, and lysofractors. And in the study, so after treating the fluorescence probes shown in this table, on the 3D RPTECs, and then 21 Z-stack images were gathering and then collected per field of view, and the maximum fluorescence intensity at the location in the Z-axis was collected to construct a single image. Here is an example of the imaging-based assessment. We selected the cisplatin and cyclosporine A, which reduced the intracellular ATP level at day 7 and no alter ATP, no change compounds in the ATP levels. So in the cisplatin treated groups, this is a pharyngeal, a marker for the cytokeratin staining. Pharyngeal intensity was decreased in the this is a concentration-dependent manner at the day 7. On the other hand, exposure to the cyclosporine exposure of cyclosporine to the 3D RPTECs, pharyngeal intensity was largely not changed, but the formation of the pharyngeal was changed, observed. So to evaluate the drug-induced proximal tubular injury using imaging analysis, we set up the evaluation method to determine the presence of or absence of toxicity from the fluorescence intensity calculated by the high-content analysis. So assuming the fluorescence intensity, the fluorescent probe in the non-drug group was normally distributed here, like here. And so we considered an abnormal state outside the mean plus minus SD value, so that we calculate the 95% of confidential area. And region defined about the 5% as toxic. And in drug addition group, the lowest concentration outside the mean plus minus SD range. And so we set this concentration over the confidential area as a LOIC, lowest observed adverse effect concentrations. It was scored within 100, within the Cmax value of 100 times Cmax is 1, and 10 times, within 10 times is 2, and scored within 1 Cmax with 3. And so we calculate the we evaluate the potential of DIKI using these fluorescence probes. So using both positive and negative compounds, imaging-based analysis was performed. And many of the positive compounds showed the alternations in the mitochondrial staining detected by the mitochondrial marker mitotracker. Predictive performance of this marker was about 81.8% for the sensitivity, and specificity is the same value. These findings suggest that the mitochondrial involvement may play an important role in the toxicity mechanism of the compounds evaluated. And other markers, so several compounds increase the Hex intensity, but no increase in the other compounds. So I have no definite conclusions. So this is other stories. So in Japan, health incidents occurred in before 2 years ago, in which patients who had taken red yeast rice supplements developed kidney injury. The Ministry of Health, Labor and Welfare reported that the cause was nephrotoxicity induced by the puberulic acid. This is a concomitant mitotoxin in the red yeast rice supplement. So, however, the compound induced kidney injury is depend on the animal species. So we evaluated the cytotoxic effect of puberulic acid in the 3D human models using the 3D RPTECs. So when synthesized, the puberulic acid was administered to the 3D RPTECs for up to 7 days. So the intracellular ATP levels was decreased in the concentration and time-dependent manner. The EC50 decreasing concentration of ATP value, 50% decrease in concentration with at day 7 was about the 25 micromolar. The EC50 value for the cisplatin is about 10 micromolar. This is also nephrotoxic, strong nephrotoxic compound, maybe the strong compounds. Because puberulic acid contains the carboxylic acid groups in the structure, so puberulic acid, we hypothesize that puberulic acid is transported by the OAT1. So we co-treatment with probenecid OAT1 inhibitor, so with puberulic acid. As a result, co-treatment with puberulic acid and OAT1 inhibitor probenecid attenuated the ATP depleting effect. So these results suggest that the OAT1 play a role in the uptake of puberulic acid into the RPTECs. Furthermore, so we imaging analysis was performed during puberulic acid exposure using the mitochondrial staining probe mitotracker. As a result, so time-dependent alteration in mitochondrial distribution was observed, along with a progressive decrease in mitochondrial staining intensity. These findings suggest that puberulic acid may induce the renal toxicity through the mitochondrial impairment. Furthermore, in clinical observations in human kidney specimens, for glycogen accumulation and decreased expression of GLUT2 was observed in human kidney proximal tubular segments. So we evaluated the effect of the puberulic acid on the glucose transporters in 3D RPTECs. as a result, the mRNA expression of glucose transporters were measured. Expression of GLUT2 was dramatically reduced by the exposure to the puberulic acid. This reduction was greater compared with the tenofovir and cisplatin exposure. So finally, it is well known that the expression levels of human renal transporters exhibit individual variability. Here, so I would like to investigate the factors responsible to the such variation. So we first performed the comprehensive proteomic analysis using the human kidney samples. So when we examine the proteomic analysis, we get the about 4,000 kinds of the proteome data for the membrane transporters. First, so we correlated with the expression count of the OA transporters, such as this is representatively the OAT1, OCT2, and CGT1 and SGAT2 correlated with the EGFR. So but the OCT2 was showing the relatively high correlationship with the EGFR. So but the OAT1, SGAT1, SGAT2 expression was not correlated with the EGFR. Sorry, this is commonly so used the clinical renal biomarker. So then, focusing the OAT1, we conducted the pathway enrichment analysis to identify transcription factors whose actively correlate with the OAT1 expression. The analysis suggested that the negative association between the OAT1 expression and was observed in the inflammatory cytokines, inflammatory pathways, particularly NF-kappa B signaling pathways. So therefore, we investigated the effect of inflammatory cytokines on the OAT1 expression in 3D RPTECs. As a result, the inflammatory cytokines, interleukin-6, interleukin-1 beta, TNF-alpha, and fibrosis cytokines, TGF-beta, so reduce the mRNA expression of OAT1, OAT3, and OCT2, but not changed by the interferon gamma. Because tenofovir is taken up into the RPTECs via the OAT1, a reduction in OAT1 expression was expected to attenuate its cytotoxicity. Therefore, after exposing 3D RPTECs to the inflammatory cytokines, we tested the cells, treated the cells with the tenofovir and quantified the intracellular ATP levels. As a result, tenofovir reduced the intracellular ATP levels by about 40%, whereas this ATP depleting effect was abolished in the treatment with the interleukin-1 beta, TNF-alpha, and TGF-beta. This result is compatible with these reduction of mRNA reductive effect of the inflammatory cytokines on the OAT1 expression. Furthermore, we examine the reduction in OAT1 expression affects the transport activity. When we the uptake of PAH, this is a typical OAT1 substrate, was measured. The decrease in PAH uptake was observed after the TNF-alpha treatment. These results suggest that the TNF-alpha treatment reduced the PAH activity. and not only the expression level with transporters. So to investigate the mechanism underlying the cytokine-induced downregulation of OAT1, so we examine the expression level of transcription factors, HNF1 alpha and HNF4 alpha, both of which are involved in regulating OAT1 transcription in the kidney. As a result, inflammatory cytokines, interleukin-1 beta, TNF alpha, interferon gamma, TGF beta, reduce the expression of these transcription factors, especially interleukin-1 beta was significantly reduced the TNF alpha and 4 alpha levels. So these results may suggest that the reduction of HNF1 alpha and 4 alpha levels by the inflammatory cytokine induce the reduction of the OAT-MR expression levels. Okay, this is the last slide. So taken together, so these findings demonstrate that 3D RPTECs, when evaluated using intercellular ATP levels, serve as a highly predictive model for the evaluating drug-induced kidney injury. And domestic ring study of the cytotoxicity test using 3D RPT obtained high inter-laboratory reproducibility. Furthermore, imaging analysis may help the understanding the mechanism of drug-induced kidney injury. So we will continue to validate their usefulness in drug and chemical safety using the 3D RPTECs. In this study, we received valuable support from many collaborators. I would like to express my sincere thankfulness to all of them. Thank you very much.
Thank you very much, Dr. Arakawa. That was a great presentation. We have a couple of questions in the Q&A. We have a few minutes for questions, so if anyone would like to add a question, please open up the Q&A and put that in there and we'll get it answered. So the first asker has two questions, actually. Do you see the question, Hiroshi? I can read it out to you.
First question.
So it says, when you mentioned the 3D RPTEC injury model at the beginning in comparison to two-dimensional, how was the injury assessed? Was it with ATP or something else in addition?
Okay, so I measured not only ATP levels but also the mRNA expression and some of the ELISA
Okay.
proteins, renal cytotoxic markers. So that's for example, cisplatin reduce the dramatically cell damage. So the mRNA expression and secret protein expression is variable.
Okay.
So finally, we focus on the ATP levels.
Okay. Thank you. And you sort of answered this question, I think, already, but can we use other viability index as well as ATP content?
Sorry, can we use the bio? Sorry, I didn't check the viability assays other than the ATP levels. But we observe the cell morphology by the high content analysis, sensitivity with less than the ATP levels.
Okay.
So I guess the mechanism is the ATP levels so reflects the simple cell death in addition to the ATP production. So for kidney, so ATP level is a very important factor for the physiological function.
Okay.
So ATP may be the good indicator for the
Okay.
nephrotoxicity evaluation. Yeah.
That makes sense. Thank you. Another question is, how did you compare the RNA expression rate for OAT1 between 2D cells and spheroids?
Yes. So, but in 2D RPTEC system, so mRNA expression OAT1 is very, very low. So under the detected levels, detectable levels. So this is very hard to evaluate the OAT1 function.
Okay. Another question going back up to the first questioner who had two questions. So his second question is, if I understand correctly, the spheroids increase the sensitivity for detecting Dicky. In the same study, did you compare the 2D system to the spheroids?
Yes. So the sensitivity is in the 2D systems. So sensitivity in the 2D system is about the, sorry, I forgot the exact numbers, but about the middle of 70. While the sensitivity in the 3D RPTECs is about the 80%, over the 80%. So this difference is caused of the transporter-mediated nephrotoxicity. Yeah.
Okay. We have a question in the chat, which is, in the 2D culture reference system, how long were the cells cultured before exposure?
Sorry.
Yeah, they put it in the chat and not in the Q&A, but it's in the 2D culture system, how long were the cells cultured before exposure?
28 days. Yeah,
Okay.
we use the same exposure period. Yeah.
Okay. Thank you. All right, let's see. Okay. Another question in the Q&A. Does the spheroids model have metabolic capacity?
Yes.
Yes. Okay.
So my collaborator, Dr. Nakajima in the Canada University, so published the reports. Yeah.
Okay. Great. Interesting question. For drug toxicity screening, do you think there will be too much overlap between drug-induced liver injury, DILI, and drug-induced kidney injury with 3D models? Though transporters are a big advantage for drug-induced kidney injury models.
Yes. Yes,
What
of
do you
course.
think about the
Yeah.
overlap?
So because so as long as I know that why so kidney specific toxic compounds happens a kidney injury, so
Right.
drug transporters has some of them drug transporter is the answer.
Right.
Substrate specificity. Yeah.
Yeah, that's true. Okay. Okay. Do you have any idea why the 3D system is not detecting all the positive compounds? What do you think is missing?
This is a very difficult question. So missing compounds is a cyclosporine and tacrolimus. These compounds are in vivo study observed in the vascular region. So some papers imp, says the cyclosporine and tacrolimus attack the proximal tubular, so we set the as a positive compound. But the cyclosporine and tacrolimus may act at first the vascular cells and then act the proximal tubular. So maybe it's a negative compound is, um, sorry, I have a exact answer. Sorry. Yeah.
That's okay. That's what, it's always what we need to figure out, right? To solve the challenges. Okay, well, this is all the questions that I see. I just want to say, it looks like on your last slide, you said that there was a ring trial ongoing. Do you think that this will be something that the regulatory agencies will want to pick up to use for screening drug compounds?
It's may prefer may prefer. So yeah. So now
Okay.
I hope to apply
Yeah.
this screening system to for example, OECD test guidelines.
Yeah.
But yeah.
That would be great. Good luck with all that work. Okay, well, thanks everyone for joining us. Thank you so much, Hiroshi Arakawa, for your presentation, for sharing your work with us. As we mentioned, the recording and it will be available on the ASCCT website. Thanks so much to Dr. Kanda for arranging the webinar and joining us today. And thanks for Allison in the background, supporting all of the work that happens behind the scenes. Thanks to you all for listening, and I wish you happy holidays because I don't think we'll have another ASCCT webinar until the new year. So hope you have a lovely holiday season. Thanks so much for listening, and everyone take care.
Thank you very much.
Thank you. Bye.